A bright field microscope is a type of light microscope that uses transmitted light to illuminate the sample. It is the most common type of microscope used in biology and medicine. Key characteristics. Uses transmitted light. Sample is illuminated from below. Image appears bright against a dark background. Best for viewing stained samples or samples with natural pigmentation. Commonly used for routine microscopy, histology, and microbiology. Dark field microscopy is a type of microscopy that uses a specialized illumination technique to enhance the contrast of the sample. Key characteristics. Uses scattered light. Dark background, bright sample. Enhances contrast and visibility of small details. Non-invasive, doesn't require staining. A phase contrast microscope is a type of light microscope that uses a special technique to enhance the contrast of transparent or translucent samples. Key characteristics. Phase contrast microscopy uses transmitted light to enhance contrast. No staining or fixation is required, making it ideal for live cells. It reveals subtle details in sample morphology, like cell membranes and organelles. Best suited for viewing live cells, microorganisms, or transparent samples. A type of light microscopy that uses fluorescent dyes or proteins to label and visualize specific structures or molecules within a sample. It works by exciting the fluorescent molecules with a specific wavelength of light, causing them to emit a longer wavelength of light, which is then detected by the microscope. Key characteristics. Uses fluorescent dyes or proteins to label samples. Excites fluorescent molecules with specific wavelength of light. Detects emitted light to create high contrast images. Allows for specific targeting and visualization of molecules or structures. Commonly used in biology, medicine, and research to study cells, tissues, and biomolecules. Thanks for watching. Like, subscribe and share my YouTube channel. @Maimuna218